becoming a group leader there in 2011. She returned to Berkeley and joined the physics and molecular and cell biology departments in 2016, and now serves as the Luis Alvarez Memorial Chair in Experimental Physics. Na has made major advances in sharpening microscopy images using adaptive optics for the study of the activity of neural circuits in the cerebral cortex. Today, Na will tell us about her work in imaging the brain at high spatiotemporal resolution. Please welcome me in please welcome Na. Thank you.
Yes, but for those few of you that may not think about brain every day, I just want to remind you that even though this is an organ that is not very big, so for human, our brain is about 3 pounds in weight, about 1,000 cubic centimeters in volume, but that's where all our mental activity is generated. And brains are made of cells that are called neurons, and neurons have different shape and sizes, but almost all the neurons will have a part which we call soma. So the one part of the neuron is called the cell body of the neuron, we call the soma. But emanating out of the soma, there's those very fine processes, which we call neuronal processes, and they can be further subdivided into dendrite, which are the processes that are receiving information, as well as axon, that are the output processes. And as most of you know, that neurons communicate at a structure that is called synapses, that are made between the presynaptic axon or specialized structure called the bouton, and the postsynaptic part on the dendrite. And if it's excitatory, then it's called dendritic spines. And neurons use electrochemical signal to communicate with each other. If you put an electrode into the membrane of a neuron and then measure the membrane potential, you will see that at rest, the membrane potential is about minus 70 millivolt. But then when these neurons start to receive input, its membrane potential will start to increase until it passes a threshold value. At this time, you will have a very rapid change of the membrane potential, which is called action potential, that can propagate along the axons and invade into the presynaptic bouton, where it will trigger the release of neurotransmitters, which will then get detected by the postsynaptic structure and then lower the membrane potential of the postsynaptic neuron. And this process can keep going, and this is how our brain processes information. Now, it is very difficult to understand the brain, not less because there's so many of the neurons inside our brain. Just for comparison, there are about 100 billion stars in our galaxy, and they are about the same order of magnitude neurons in our brain, and those neurons make another 4 order of magnitude connections among them. Even when you study a much smaller brain, for example, that of a mouse, there's about 100 million cells, 100 million neurons in the brain, and about, again, 100 billion connections that are made between those neurons. And what makes it difficult to understand the brain is because of the mental activity. Everything that is going on in our brain is not the property of a single neuron or a single synapse. Instead, it's really an emergent property of many, many neurons working together in a circuit or neural network where they were able to work together through the connections, the synapses between them. As a result, if you want to understand the brain, it's not enough to study a single synapse or even a single neuron. Instead, you need to have a kind of method that will have very high spatial resolution, ideally a sub-micron spatial resolution, which would allow you to study the processes that are happening inside individual synapses. You also would like to have a method that has millisecond time resolution so that you can keep track of the action potential generation, which is on the order of millisecond. And this method should be able to study structures that are at depths, millimeters down into the brain. And also it should work in a sample that is optically opaque or optically scattering, for example, as you can see here. So one of the methods that hold the promise of satisfying all those conditions, if not all at the same time, is called two-photon fluorescence microscopy. So for those of you who know fluorescence, is that you have a molecule which is usually in the ground state. And then you can excite the molecule to the excited electronic state. And when it comes back to the ground state, some of its energy can be released in the form of a fluorescence photon. Now, in two-photon fluorescence microscopy, the molecule needs to absorb 2 photons at the same time in order to go to the excited state and then generate the fluorescent emission. This process has a very small probability to happen. And for those of you who are trained in physics, you need high order perturbation theory. And as a result, this process will only happen when the electric field has very high intensity, which means that it will only happen in reality at the focus of your excitation laser. So now, then how do we do the experiment? How do we take image? So with 2 photons, you can focus your excitation laser into the brain. And then at this focus position, you can record how many fluorescence photons coming out. And to form an image, you basically just scan this focus in 3 dimensions inside the sample, and then you will get a micrograph of the fluorescent molecule distribution. And as you can see, this imaging method has a type of resolution that allows us to see individual synapses, as you can see in the form of dendritic spines here. And one other nice thing is that it works in scattering sample because the signal is only generated at the focus, and you know exactly where your laser focus is. So then you can form this kind of nice images in an optically opaque brain, sample like the brain. And what makes this method very, very useful for neurobiology is the development of genetically encoded activity indicator. So here you are looking at a movie of neurons that are expressing a protein called GCAM3. Whenever the neuron fire action potential, the fluorescent signal will go up. So by imaging it, this brain, we can now convert this electrochemical signal into an optical signal that can be detected by the 2-photon fluorescence microscopy. Now, with newer version of the indicator, for example, GCAM6, which is a calcium indicator, you can even measure this process with the sensitivity of single synaptic terminal. Here is one different type of dendritic spines that are activated by different type of sensory stimulus. So this looks great. We have a method that works in scattering sample. It has a very high spatial resolution. And we have the probes that would allow us to monitor the activity of the neurons in vivo. What is the problem? The problem is that you don't get such high resolution and good quality images anywhere inside the brain. Once you start to image deep into the brain, which are millimeters in dimension in the case of a mouse brain, then the optical inhomogeneity of the tissue itself start to degrade your image quality. So ideally, when you do 2-photon excitation fluorescence microscopy, what you want to have is to have a very tight focus, right? As small focus as possible, which require all your light rays to go to the same point, which would be your focal point. And they should also have the same phase so that they can constructively interfere and give you a very tight focus. But once you start to image into a biological sample, the optical inhomogeneities change the direction of propagation for your light rays so that they do not overlap at the same point. And where they do interact, they don't have the right phase relationship to give you a very tight focus. So as a result, instead of very small and bright focus, now you will have a dimmer focus as well as an enlarged focus, which means that now you will be losing both signal because the intense signal is proportional to the intensity to the second power, and you will also lose resolution because now you cannot confine your excitation to as small a volume as possible. In other words, whenever you are moving beyond the cultured cell on a cover slip, the diffraction limited resolution, which would dictate the size of the focus here, is actually not that often achieved, even no matter how good the microscope you have. That's because your sample itself degrades the image quality. How do we deal with that? If we know how the sample is distorting the wavefront of the excitation light, you could actually pre-distort it in the opposite direction before you even put in the light into the sample, so that it will cancel out the sample-induced distortion and recover a diffraction-limited perfect focus inside the sample. So this kind of method is based on the concept of adaptive optics. And for this talk, unfortunately, I don't have time to go through all the detailed method of how we actually measure this aberration and then correct it. It's just to say that we already generated multiple different methods to do that. And you could, if you're interested, ask me a question later on or go to those papers. But I just want to explain to you on the conceptual level that adaptive optics that we do for microscopy was actually very much inspired by what astronomers has been doing for many, many years when when they try to image a star using a ground-based telescope. So ideally, you want the same thing happen, that the starlight will be focused nicely into a focal spot onto your detection camera. But before that could happen, your light has to pass through atmosphere, which again distorts the excitation light, just like the biological sample distort their excitation light. Instead of a very sharp image of the star, you have something that is very fuzzy. But if you can similarly measure the aberration of the distorted light, then you could correct it, then you will have a much sharper image of the same star. Here is just an example with images of the middle of the very center of our galaxy, measured without and with adaptive optical correction. So you can see there's a very drastic improvement of image resolution and the contrast. So it turns out that the method that astronomers use to correct and measure and correct aberration cannot be directly translated into biological sample because our sample is optically opaque. But then we developed a set of methods that would allow us to do this. And here I'm just going to show you a few examples. So here are the images of dendrites at about 600 micron below the surface of the brain, measured with the help of adaptive optics. So you can see the dendrite, you can see those little protrusions, which are the dendritic spines imaged very clearly. But if you do not use adaptive optics and look at the same structure, you can see that the image quality is much worse. Even though in this case, we're already doing absolutely the best conventional optics can do. So here we're using a microscope objective that has a correction corner or correction ring on top of it, which can partly correct for sample-induced aberration. But you can see that without correcting the full aberration coming from the sample, it still cannot give you very nice single synapse resolving resolution. Here I'm going to show you next. So what is this good for? Can you actually discover new biology by taking good imaging data? So one question that in my own lab that we ask is a question inside the early visual processing inside the brain. As you know, that the visual information first arrives in the eye at the retina, and from there it will go to a structure called thalamus, or in this particular case called LGN. And from there, the thalamic neuron will project to the primary visual cortex of this sensory information. So we ask a very simple question: what kind of information is being sent to the visual cortex from thalamus? And we can answer that question using imaging by transfecting the neuron in the thalamus with this activity indicator that I mentioned earlier, the GCAM6. You can see the cell body in LGN here. And then you can also see the axon of those coming from the LGN inside this brain structure called primary visual cortex. Now this is just a confocal slice showing you how the axon distributions are. With the help of adaptive optics, we can image those axons, those botons, individual synapses on those axons called botons at very high resolution, even hundreds of microns below the surface of the brain. And you can see there's not much difference in the image quality we obtain here. And then we ask a very simple question. It is known for many, many years that the neurons inside the primary visual cortex select for the orientation of the visual stimulation that is being presenting. In other words, the neuron will fire action potential only if the grating, the visual stimulus you give to the animal is along certain orientation. Now, classically, it's also believed that such a property is only the property within the cortex instead of coming from the thalamus. They think that the thalamic input was not tuned, was not orientation tuned. But because we can measure those inputs directly now on single synapse level, we can do this experiment and ask, are part of them actually selective for orientation? And what we found is that more than 50% of those thalamic inputs are already selective for orientation. And then later on, we use a different improved sensor, which even allow us to image 600 micron below the surface of the brain, where we see the same thing, that, for example, this button 1 and 2, they only become active when the grating that we present to the animal is more or less vertical, but not active when the grating is horizontal. Interestingly, if you do the same experiment on the same sample with or without adaptive optical correction, and looking at the tuning curve of individual synapses, you see that for some of those botons, the differences is small. You can see that adaptive optics, which are the black curves, sharpens the tuning curve a bit. But even before the correction, you could tell that this button is orientation selective. But for many more botons, the difference is much more drastic. Without using adaptive optics, you would think that those botons are not really tuned. But once you use adaptive optics, the tuning curve becomes much sharper, and you can say, unambiguously that those are the tuned input. Now, the mouse has no way in knowing whether we're using adaptive optics or not. So it's not like it's trying to mess with us to say, hey, I'm going to have a sharp tuning now that you're using a better microscope. So something is more fundamental is really happening here. It's actually something very, very simple. So those are the botons. It's just a cartoon of botons that, so this is like imaging depth in this axis. And because this is a mouse, it does not have this so-called orientation tuning column. So the botons in here actually has different tuning, different preferred orientation. So when you do not use adaptive optics, you will have a big enlarged focal volume. And even though you think you are imaging the response of the button that's at the very center of your focus, but your signal is now actually contaminated by the structures above and below the button that is right in the middle. As a result, because they have different tuning property, as a result of this averaging, now you will have a broadened tuning curve. But once you use adaptive optics to shrink down the size of your focus, then you will much more effectively reject those contaminations, which otherwise will basically give you the wrong tuning property. So this is really where adaptive optics make a huge difference, is when you have this kind of very dense innovation and they carry different type of information. And if you do not have adaptive optics, you will reach the opposite conclusion that the synapses are not really tuned. So to image even deeper, so with two-photon fluorescence microscopy, we more or less can image the first 1 millimeter of the brain or the entire cortex. But if you want to image even deeper, and if you are trained as a physicist, you know that when the light wavelength goes longer, the scattering of the light goes down. So instead of using two-photon fluorescence excitation, you could use three-photon fluorescence excitation, which would allow you to penetrate further into this scattering sample. And this method, three-photon fluorescence microscopy, is pioneered by Chris Schi's lab at Cornell University, where they show that by using three-photon excitation, you can image another 500 micron into the brain. So we combine adaptive optics with three-photon fluorescence microscopy. And here is an example of how it helps to improve the image quality. You're looking at a neuron that is at 750 micron below the surface of the brain, measured without and with adaptive optics. You can see there's a huge difference in the signal intensity, a difference that is bigger than what we saw with two-photon excitation. Because with a higher order nonlinear process, it's even more sensitive to any kind of aberration. So you can see that for this cell body, we increase its brightness by almost 7 times. And for this fine dendrite here, we improve its brightness by 19 times. And we can zoom in further at one corner of this image. And again, you can see that the difference is really big. And for this stretch of the dendrite, we improve the signal by 16 times. And if we zoom in even further and look at this very fine dendrite here, and on the left-hand side is without adaptive optics, but we artificially increase the brightness 10 times, so you can see what the heck is going on there. You can see there's some faint hint of dendrite and possible dendritic spines, but you wouldn't be able to say those are dendritic spines if you don't know, with the help of AO, that indeed there's a dendrite, the dendritic spines, and we can even see very fine spine neck. So as you can see, that adaptive optics really very much improve the image quality in vivo. So more recently, we also did a project that is not my typical work, because I'm not really into the super resolution imaging field. But more recently, I had a very courageous postdoc who want to do super resolution imaging in vivo in a living, breathing animal. So it turns out that, unsurprisingly, that adaptive optics is also essential when you try to apply super-resolution imaging in vivo. The super-resolution imaging module we choose is called structured illumination microscopy, where you use structured light to down-modulate some spatial frequency into the band pass, the pass band that can be detected with the diffraction-limited detection system. I'm not going to go into detail, but just to say that the challenge of applying this structured illumination method in vivo is that we have now a three-dimensional sample, and you will have out-of-focus signal, and of course there will be the problem of sample-induced aberration, and then finally there's also the problem of motion, because the animal is, there's a breathing, there's a heartbeat-induced artifact. When you have a resolution around 100 nanometer, while your brain is moving, 200 nanometer, how do you get the super-resolution images? But Ralph managed to solve all those problems, and just to show you the super-resolution performance of this structured illumination microscopy, we image the same structure using wide field, deconvolved image, and also 2-photon, which is a standard in vivo imaging method, and also structured illumination. And just you can clearly see that this method has a super resolution as it can resolve structures that cannot be observed with the other method. You can quantify those images, for example, by comparing the size of the neck of the spine. As you can see that the SIM gives you the highest spatial resolution, or we can look at internal structure here. Again, SIM was able to resolve it, but you can't really see it with the other method. And as I mentioned, unsurprisingly, it is very sensitive to the presence of aberration. So if we take an image without the adaptive optical correction, you have all kinds of imaging artifacts. For example, instead of seeing a spine coming out with a very fine spine leg connected to the dendrite, you have this very smeared out images. And in another example, here is a spine head. Without the adaptive optical correction, you see this kind of weird ghost image of the spine head. But after correction, now you see a very sharp images. Just to show a few more in vivo example. So here again are two examples. On the top is an example where the cell is labeled with a membrane marker. And you can see that the high resolution of SIM allow us to see the hollowness of the spine head, which you cannot really see with the diffraction-limited method. At the bottom is another example of a cytosolic expression, so where the protein is just expressed in the cytosol. And again, you can see those internal organelles, which you couldn't really see before correction. So this is really just kind of a little side project in the lab, just to show that adaptive optics is really something absolutely essential, whether you work on diffraction-limited method or super-resolution method, in getting the proper images in a real biological in vivo type of sample. But coming back to my interest, which is really more on the neuroscience and circuit level, then now that we can image things at very high resolution, the next thing that I was telling you before is that imaging a single spine is not going to really tell us too much about how brain works. We really need to be able to imaging activity over a large volume of many, many neurons at the same time. So as I said, that with adaptive optics, we can imaging the first 1 millimeter of the brain, which in the case of the primary visual cortex, it means that we can imaging all 6 layers. This is a layered structure. All 6 layers, we can image them at very high resolution. But how about imaging all those neurons simultaneously at very high resolution, both temporally and spatially? Now that is a challenge. Why is that? The challenging is because with the 2-photon, remember I was telling you the way we form an image is by scanning this focus. I don't know whether you can see. Hopefully you can see a little bit. You can see the focus is scanning, right? When you scan it in 2 dimensions, that will give you a 2-dimensional image, a very optical, what we call optical section of the image, which is really just a very small part of this structure. In order to get a volumetric information, because brain itself, the circuit and the neuron itself is really a 3-dimensional structure, you have to now scan your focus in 3 dimensions. And very obviously, it's going to take much longer, right? If you have 10 Z sections, now your imaging speed will be 10 times slower. So how can you image at a volume at a very high speed? It turns out because in the brain, nothing is moving. Unlike, if you study a protein that is moving around in the cell, you want to resolve the structure always in 3 dimensions. But in the brain, nothing is actually moving around during our time of imaging for the structure that we're interested in. So you could actually stretch out the focus to make it really, really elongated while still maintain the natural resolution. And this is what we call a bezel-like focus, or bezel focus. And just in contrast, the traditional way, we call them the Gaussian focus. But the nice thing of using such an elongated pencil beam to do 2-photon excitation is that by scanning this structure in 2D, you will get a projected view of the entire volume. And now your frame rate, in this case, if you will image at 30 frames per second, now it becomes 30 volume per second. And you can imagine the structure at different depths simultaneously and interrogate their activity at a very high temporal resolution. So here is an example of what type of biological question that you could use this method to answer. For example, again, going back to the visual cortex, if you want to know what are the synaptic inputs onto an individual primary visual cortical neuron and what kind of preferred orientation those inputs are, now in a conventional way, you can see that this is what the dendrite looks like in 3 dimensions. So it's a very much 3-dimensional structure. To image across a volume of only 60 micron, it actually will take, with the experimental protocol we are using, it's going to take 10 hours to do this experiment, which is really just not practical in reality. But with bezel beam, you can now interrogate all the dendrites and dendritic spines in this volume in a single 20-minute experimental session. So I apologize, the movie is kind of compressed too much, so it's kind of fuzzy, but hopefully you can still see the isolated onset of the activities on those individual dendritic spines. And we could look at, we can analyze this data and identify the spine-specific input. For example, this spine is only activated at this particular orientation, but the nearby dendrite do not have any activity. And then we can ask the question, what are the preferred orientation of all the spines in this volume? And in this way, we can answer this type of question at a very high throughput fashion. Now, of course, we don't need to limit ourselves to the interrogation of individual synapses. As long as the sample labeling is sparse enough so that you don't have too much overlap in your projected image, you could interrogate all of them in the volumes at the same time. So one example are the inhibitory neurons in the primary visual cortex. So the inhibitory neurons are the neurons that would tamp down the activity. And if they are not active enough, then you may have problem like epilepsy. So you can see it's about 10 to 20% of the cortical neuron population. And the movie starts to play, but let me see whether I can have them. Anyway, so you can see that in each individual slices, you only have a few neurons that are inhibitory. And they're so few that even when you look at the projection of 100 micron thick volume, you don't see really much overlap between neurons at different depths. This means that in order to interrogate the first 400 micron of the cortex, we do not need to take 80 images at 5 micron per step. Instead, we only need to take 4 images where 100 microns of them was interrogated at the same time. And now instead of only a few neurons, you will get the activity of many neurons at tens of neurons at the same time. And when we were doing this experiment, we didn't really know what to expect. But when we get this calcium transients out and we look at it, we notice that actually a subpopulation of them has very highly synchronized activity in that they are active at the same time. And this synchronized activity actually has nothing apparently to do with when we present the visual stimulation. So then we decided to interrogate it a little further by looking at the different subgroups of the inhibitory neuron. So we then labeled the neurons that are called VIP neurons, which is even sparser because of the subgroup. You can see within 100 micron, there's not that many of them. But then when we do the same experiment, we notice that almost all, except this one, almost all the inhibitory VIP neuron has highly synchronized activity. And it turns out that my postdoc was doing something that we, again, wasn't really know why we were doing it, but he was just monitoring the pupil diameter of the mouse. And then what we saw is that this spontaneous activity is very much synchronized with the pupil diameter. And this is just the correlation coefficient matrix. You can see that it's extremely highly correlated. Now, what does the pupil diameter tells us? When you're aroused, when you're interested in something, when you're very much awake, or when you drank too much coffee, your pupil diameter will increase. So hopefully, all of you guys' pupil diameter is dilated at this moment. And if not, then just relax. I know it's lunchtime. That's fine too. But basically, what we see is that this VIP interneurons has highly synchronized activity. That is really just reflecting the arousal level. So if you're into the inhibitory neurons, the next obvious experiment is to look at another population of inhibitory neuron called somatostatin neuron, because there has been work published suggesting that the VIP neurons inhibit the somatostatin neuron. Now, if that is true, you will expect that the somatostatin neurons activity should be anti-correlated with pupil, right? And indeed, we find somatostatin neurons that are anti-correlated with pupil, such as those blue traces. But we also find a substantial fraction of them that are also correlated with pupil. What this tells us is really like this particular transgenic line don't just label somatostatin neurons are not a homogeneous population. They really have a very heterogeneous type of activity. And there's actually, I think, more recent work suggesting that indeed that is the case. So I'm not really trying to tell you any biology here, but I'm talking about this is just because our ability to look at many neurons at the same time allow us to easily see something that wasn't already in your hypothesis. And this is the power of imaging. This is the power of high throughput imaging of many neurons at the same time. So I just want to quickly, before I move on to the next data set, to say that, so obviously there's a problem when you want to image very deep structures in the brain that is opaque, is that the optical scattering will just kill all your signal and your excitation. So that you can really imaging using a conventional method, a structure that is 5 millimeter down, inside the brain, even without using 3-photon, 1 millimeter down in the brain. Now, one of the methods that can allow us to do that is to use what we call a green lens. So it's basically a very miniature optical component that can image the focal plane of a conventional microscope objective onto the sample. So like image relay. So what we found that we can generate those bezel scanning even through those green lenses at a very high resolution. So here just some in vivo example where we're looking at a mouse hippocampus. And on the left-hand side, again, are the Gaussian images where the structures are color-coded by depth, I should have mentioned for my previous slides as well. And with the bezel beam, we can basically, instead of having to take 15 images to cover this volume, with the bezel, we only need to take one image, and we can see all those neurons cell body very easily in the single scan. And when we zoom in, look at the neuronal processes, what you can also see is that we can see axons, axonal boutons, dendrites, and dendritic spines. I'm not going to go into the detail, but it turns out that the bezel beam actually has a higher lateral resolution than the conventional imaging method. So in all these examples, you can see like those dendritic spines looks a little bit sharper and more easily distinguished compared to the conventional imaging method. So in addition to this higher throughput, in terms of the lateral resolution, we also even have a gain of the lateral resolution. So with these approaches working well together, so you can really be able to do this very high lateral resolution imaging and high throughput imaging at the same time. So more recently, we combined the bezel imaging module with a microscope that was built by my former colleague, Carol Svoboda's lab, what they call a mesoscope. A mesoscope is basically a 2-photon microscope with a very large field of view. So this is what the microscope looks like. It's very much customized, but it has a 5 millimeter field of view where you can do 2-photon image in it. And we were able to design a very small module that we can then attach to the existing mesoscope in Carol's lab. And you can see the module is only on this bit of it. It's about a 12-inch by 12-inch optical plate that we attach to the existing system. And then we can then just scan a single 2-dimensional scan. This allows us to image a very large field of view. But the nice thing about the image using bezel is, of course, that now a single scan gives you a volumetric information. So just to zoom in on this image, and you can see if we zoom into this part, you can see now lots of dendrites. If we zoom in even further, you can see dendrites, dendritic spines, you can see axons, axonal boutons. So now we have a way to do this synaptic level resolving imaging over extra large volume. One other thing that this method has been super nice to do is also allow us to look at all the dendritic input onto a single neuron simultaneously. So here is just a conventional image movie of a cell body. It's a rather boring movie, so I'm not going to show you because this neuron has a cell body at around 600 micron, and it sends its dendritic tree all the way to the surface of the sample. Now, using a conventional imaging method, you need to take 300 images to cover this volume. But with Bessel beam, we can cover this volume in 6 scans of 100 micron long Bessel beam. And you can still see that we can resolve individual dendritic spines that you could see there, right? But the really improvement, of course, is temporal resolution. Now we could image the entirety of this neuron at 3.4 volumes per second. And you can, in this case, it's just spontaneous activity. The animal is awake, sitting in the dark, and just a spontaneous activity that is happening there. And we can identify all the dendritic spines, and it turns out it's 145 of them, actually. There's a typo there. And you can see that those spine calcium signal has all this synchronized activity. That is because the cell body is firing action potential, and the action potential backpropagated into the dendrite. So this is what we call the global event. But for many of them, you will also see local synaptic input that are not synchronized with all the global events. And those are basically what people will be very interested in, is what are the synaptic input that is being received here. Now, with this method, we finally have a way to answer this question: is what is the input-output relationship of a single neuron, which requires to interrogate and look at all the spines or synaptic inputs onto the single cell, which is often in 3 dimensions. But with Bessel beam, now we can really interrogate this at the real time in the context of a calcium imaging, which is, of course, slower than the membrane potential. But we will get to the membrane potential part later. So with the mesoscope, of course, the biggest advantage is this very large field of view. So here we are labeling all the, again, inhibitory neuron in multiple cortical region. So here is just the imaging stack, but separated into 6 regions, goes from 50 micron below the surface of the brain all the way to 650. So each colored dot is an individual inhibitory neuron here. And the volume is basically 1.4 millimeter by 3 millimeter by 600 micron. So we can now look at the activity of those inhibitory neurons at the same time. Again, this mouse is not actively engaged in anything, it's just sitting in the dark, but by no means its brain is silent. You see all this spontaneous activity of those inhibitory neurons. And if you're staring at them carefully for long enough, you also have the kind of almost like illusion of waves going, from different regions. But then, of course, we have very high lateral resolution. So in this case, in this data set that is about, I think, 30 minutes long, we identified about 9,000 inhibitory neurons. And here I'm plotting all their activities ranked by depth, as well as how correlated they are with the pupil diameter here for you. So you can again see there's a population of inhibitory neurons that are positively correlated with pupil diameter. There are populations that are negatively correlated with pupil diameter. And just to show you, we can recall them all the way to layer 6 and find all those neurons. And we can color code each individual neuron by their correlation coefficient with the pupil diameter. And I'm not going to go into the detail of the biology because we only have n equals to 1. This is what technology can do. You only need 1 example. With biology, you need to have a lot more animal. But just in this even single animal, you can see some differences in different cortical regions. From the more posterior to a more anterior region, you can see there's a little bit more positively correlated neuron with the pupil diameter. And imagine that if you are interested in particular behavior task and you use this kind of method to measure the neural activity, you get a huge amount of information of how the brain is behaving during your task. So how about if I want to not just limit myself to inhibitory neuron? What if I want to look at all the neurons in the cortex at the same time across all 6 layers? Now we have a problem. It's because the cells at a different depth will now start to overlap just because there's so many of them. And one way to make this problem a little bit less severe is by limiting the expression of the activity indicator only to the cell body of the neuron. In this case, we are limiting their expression only in the nuclei of the cell. So you can see an imaging stack in the cortex. Still, it's fairly dense, right? So if you project this 80 micron thick volume on top of each other, you still see this quite a bit overlap. So now when we use Bessel image to image them, we cannot avoid those overlaps. Those cells are right on top of each other. But what helps us is that if you look at the imaging data, you'll notice that at any time, only a small subset of the cortical neuron is actually active. This is like a sparse activity that people have been observing in cortex for a very long time, which means that even if these two cells are overlapping to a certain degree, as long as they are not right on top of each other with no way to tell them apart, and if they are active at different times, you could actually use an algorithm to separate these things. In a way, it's like the super-resolution localization microscopy, because if the molecules are fluorescent at different time, you could localize them, even though they are very close together. So in collaboration with Leon Panitsky's lab at Columbia University, who developed those called constraint non-negative matrix factorization method, we was able to pull apart the overlapping neuron. Now this is using the algorithm that was not optimized for this type of data, but even there we could pull apart overlapping neurons and determine their orientation tuning property. But since then, we have also Leon's group has optimized their algorithm for the type of data that we are looking at. We are also collaborating with Betty Hung and David Anderson's lab in Caltech to apply this method, try to do a whole brain imaging in the case of fly and whole cortical region imaging in the case of mouse. So this is still ongoing, but I just want to show you one example in the fly. Here you are looking at the canyon cells in the olfaction, the mushroom body, the olfaction region of the fly brain, where the expression are limited to the nuclei, similarly. So you can see that those neurons are kind of very much in a three-dimensional structure. The different cells are at different depths. Conventionally, it would be very difficult to image them. But with Bessel beam, of course, we can image all those structures simultaneously. And Betty's lab actually came to us, brought their fly with them, and then they were giving the same fly 3 different odor, and you can see that the Bessel beam can pick up on the activity of those neurons. And using the optimized program coming from Leon's lab, we could also pull apart those overlapping cell population automatically. In this data set, we identified about 300 active neurons. That means these are the neurons that were activated by at least one of the 3 odor, which is very much in line with their prediction of what is the population of a canyon cell that will become active. So hopefully with this, we will be able to do the same type of mesoscopic imaging, but now with all the neurons inside the brain and looking at their activity when the animal is engaged in a complex behavior task. So finally, before I end my talk, I just want to say that now I've showed you that with adaptive optics, we can have very high spatial resolution, even deep inside the brain. We can image them at depths. We can image them at pretty high temporal resolution that is sufficient for us to capture all the calcium activity, which is nevertheless it's a kind of a low filter, a slower version of actual activity event. Now, as I mentioned at the very beginning, the action potential happens on the level of millisecond scale. And it's really more desirable if we could image those action potentials or the membrane potentials at 1,000 frames per second. And as I mentioned to you before, obviously this is going to be difficult, right? It's even harder just to do the 3 dimension, and then we can do 30 hertz. But now you want to increase the scan speed by another 30 times. How can we do this? So for many years, this wasn't really a simple solution. But until I was going to an imaging conference, Photonic West, and I met a guy's name who is Kevin Shah. So he's a professor at the University of Hong Kong. And he actually developed last year, or 2 years ago now, a method called free space angular chirp enhanced delay or FACET. So he developed this in order to image cells that are flying by very rapidly in microfluidic channel. But it turns out that this method can be very easily combined with 2-photon fluorescence microscopy. So we can imaging neurons at very high speed. Now, what is this method about? So I have to admit to you that when I look at the name, even though I'm trained as a physicist, I have no idea what this method is. But the concept is actually very simple. So as I mentioned to you before, for 2 photons, the way we form an image is to send a laser, and it's always a pulse laser, and through the objective into the brain, and you will have a galvo scanner to scan this focus to form an image, right? So what is a facet module is very simple. It's made of a cylindrical lens and a pair of mirrors that are almost parallel to each other, except for a very small angle alpha. So when you have a cylindrical lens, you will now focus this light down in 1 dimension, and then you can then send this into the facet module. So now something interesting is going to happen. If you look at this red beamlet, okay, so what it's going to do is that it will bounce, bounce, bounce. If the 2 mirrors are perfectly parallel, it will just go all the way out, right? But because of this very small angle, the incidence angle onto those mirrors keep decreasing until at one state, on one of the mirrors, now the beam will be perpendicularly incident. And when that happens, it will be bounce back. And then if you have a beam splitter here, you can then send this beam back through the microscope and form a focus inside the mouse. Okay, so why do you go through the trouble to do this? The interesting thing is that now if you look at a different beamlet, for example, this dark gray one, because of this cylindrical lens, now it enters this mirror at a slightly different type of angle. So as a result, in this case, because the angle is smaller, it will reach the normal incidence a little bit earlier. So when that happens again, it will come out. It will come out at a different angle, right, because it's back retroreflection. And the different angle means that now it will form a focus at a different location inside the brain. But not only that, one way of looking at this is that you have a focal spot here. And by different bounces of different number of bounces on the mirror, you will generate different virtual images of this focus. And this different focus will be at a different distance away from there. In other words, that they are temporally delayed. This red pulse will arrive after this gray pulse, because gray pulse don't have to travel as much. Now, it all sounds if it sounds a little bit complicated, then if you ever take a selfie, a picture of yourself in 2 mirrors, then what you will see is exactly that. Like the image there of this little girl has bounced in between the mirrors more times than the image of the girls here. And if you're more artistically inclined, there's a famous Japanese artist. Her whole career is kind of amazing. Her whole career is by putting objects between mirrors. And if only I know, right? But anyway, so she has this thing called infinity room, which is basically a lantern in this case, in between a couple of mirrors. And all those multiple reflections generated all those virtual images. And she has a very kind of depressive title of it, but never mind. But in our case, in a happy thought, is that what this facet module did with the cylindrical lens and 2 mirrors is that it separated the focal spot from the light excitation pulse into multiple pulses that each hit at a different location, right? It's like a 1-dimensional scan. And they also arrive at a different time, so that, and we basically make them delay, in our case, by 2 nanoseconds, which is like fluorescence lifetime. Then we can separate the fluorescence coming out of those individual focuses. But then the nice thing there is that to form an image, we do not need to scan in this direction anymore, because this module did it for us. We just need to scan the focus in the orthogonal direction, which means that our galvo line scan rate now becomes the frame rate. And even when you use an ordinary galvo, not the resonant type, it's very easy to scan at 1,000 lines per second. So then, Kevin's postdoc, Danny, who first joined my lab at Geneva as a visitor, but later on joined my lab at Berkeley. So he added the facet module to our 2-photon microscope. And then we were able to now actually doing the scanning at 1,000 frames per second. So to begin with, we didn't want to push too hard all the way to membrane potential. So we started to try a different type of activity sensor that senses glutamate. It is faster than calcium. And this is just one of the only, I think, cultured neuron example. Here, where we are looking at 2 different variants of the glutamate sensor and imaging them at 1,000 frames per second. You can clearly see the activity. So this is a faster variant, harder to detect. But it is really very easy. When we image at 1,000 frames per second, we're like so severely oversample this process. But just to show you that we can detect those things very, very easily because how fast we are. And we're looking in vivo as well. And here we're just looking at the spontaneous glutamate release. And in those processes, again, we see those responses very easily. And interestingly, when we were doing one experiment, we noticed when we look at this 1,000 frames per second that in this small region, there's a bright structure very rapidly just flying by through the field of view at a speed of 1 millimeter per second. Now, the only reason we were able to see them is because we're imaging them very fast. And we think it's probably like a microglia cell that gobbled up the fluorescent molecule and just flying by in a blood vessel. But now, of course, the ultimate goal is to imaging membrane potential. And luckily, we got some new variant of genetically encoded voltage sensor from Michael Lin's lab at Stanford called ASAP. So ASAP is a reverse sensor, which means that when the membrane potential goes up, the fluorescence actually goes down. And we are lucky because the sensor's brightness is just enough for us to say that we are detecting single action potential. So this is one example. This is in vivo. It's a very densely labeled sample. We are looking at this cell. And you can see that most of the time the fluctuation is around baseline. But from time to time, we see this kind of deflection that we think it's actually real action potential. It's about signal to noise ratio of 3. So now they have newer variants, which we are very excited in trying. Hopefully, we can push this higher so we can actually see it on sub-threshold event. Here's another example. We are doing a more sparse label here. And again, you can see that this deflection. And this neuron fired 157 spikes in about the same number of seconds. And if we just put all the traces together, you can see that it's pretty clear there's something there. But then, of course, am I just giving you the publication typical example? And just to say that, no. So those are all the in vivo cells that we actually image. It's about 30 cells. And ranked from the number of spikes that we detected, even when it's 8 spikes, we can clearly see those transients. And we could also fit those responses and then decide what are the rise time, fall time, and the full width half maximum of those optical spikes that we got. And those values are very much in line with what Michael's lab know about the sensor's dynamic. Of course, I'm interested in vision, so we then also, instead of just looking at the spontaneous activity, we also look at the visually invoked activity. So here is a neuron, and in primary visual cortex, and those ticks are the optical spikes that we measured when the animal was in the dark versus when we give them drifting grating stimulus. So you can see that this neuron is firing even in the dark, but once we start to give them drifting grating stimulus, about 80 milliseconds after the onset of the stimulus, then you have an increase in the spike rate. This data set looks very similar to what people get with the cell-attached recording, electrophysiological recording in vivo. And it also have the right temporal response from the onset. So we're very happy about that. And we also find neurons that also has orientation selective response in this example. So what we are working on right now is try to push the sensitivity of our method a little bit more and try to get better sensor. But something that we're very excited about doing is that with the facet module, we can change the Galvo scan rate into the frame rate. And the next thing obviously is to add bezel to it. So now we will change the line rate into a volume rate. So we will be able to imaging at 1,000 volumes per second, which will really allow us to interrogate electrical activity at the time scale that it deserves. So finally, I just want to thank the member of my lab. And this is the last picture after we pack everything up from Geneva of my lab members. And the people who contributed to the work that I described here, Yajie and Rongwen worked on the bezel imaging project, and Guanghan worked on the bezel combined with green lens, and Danny worked on the 1,000 frames per second project. Christina worked on the 3-photon AO, and Ralph is not in this picture already. But we also had lots of great collaborators, both inside Geneva as well as in other institutions. And I have to thank my funding agency, as well as a disclosure that the Bessel imaging method has been licensed to Thorn Labs. And thank you very much for your attention. Question?
For the voltage imaging, do you
Right.
still have the constraint that it has to be sparsely expressed?
No. So, well, for Bessel, eventually it would be, right? That would be a big problem. We can't really,
So you have a ceiling.
yeah, for the density. Unless you only care about cell body, then you can do the same thing. You confine the expression only at the cell body. Then, again, pull them apart when they are only overlapping, but not right on top of one another. Actually, it turns out Michael's lab does have a cell body concentrated version of it. That's something that we want to try, because he told us that that version has better sensitivity than the whole membrane labeling type. Yeah.
Great talk, thank you.
Thank you.
Can you tell us about the spatial extent that you can do this 1 kilohertz imaging?
Right.
Is there a limit? Or is
With the Bessel beam, with the Bessel beam type.
with the
Oh,
picture at the edge.
just a 2D, right.
just 2D.
Very good question. So let me see. Right. So, in the direction that we're using Galvo to scan, we can scan as large as we want because the Galvo is scanning. But in the other direction, in the facet direction, we're limited by how many pulses we can generate. So for those data, we're generating 100 pulses. So there's only 100 pixels. Now you can space them differently, but that won't really increase in your pixel count. So what we're also working on right now, because one of the reviewers is like, oh, this is not enough, right? Like if you can double it, triple it, I would accept the paper. So to generate more pulses, basically you need to use longer mirrors. And so one thing we're doing, okay, let's use longer mirrors, and you can generate long pulses. The issue here is really because we are separating the first pulse and the last pulse just by how many bounces there are, right? And you can basically calculate with 100 pulses 2 nanoseconds apart. The first pulse and the last pulse are 200 nanoseconds apart. And speed of light is 3 times 10 to the 9th meter per second, so that's 60 meters. Already. No, is that right? 60 meters from the first to the last pulse. So when you have longer pulses, you will have loss on those mirrors. But that's basically, in a way, it's silly, but that's basically what's limiting us. We have our excitation laser happens once every 1 microsecond. So we could pack 500 of them in principle. But in reality, we just don't want to have the device make them too long. Yeah.
Yes, now you have a lot of light and you need to do it at very short intervals. How fast is the ASAP3 photo bleach with how do you take video for?
Right. So let me show you the raw data, okay? It works surprisingly well. I was expecting that just gonna go down so quickly, and instead, we see this very weird thing. So this is the real data. Just to show you how we analyze the data. So there are bleaching. Okay. There are also some motion artifact because it's live animal. So we basically did a smoothing and then as a background. Then we subtract out the background, and then you can see those extra things which we think are spikes. But the interesting thing is that when you just continuously take data, we do have a stop. For example, when we're giving the animal blank stimulus, then we stop. You see the signal goes down, and then 0.5 seconds later, we scan again, it goes back up again. So several things are happening. One thing that's really, the people who work on fluorescence protein actually say there's some kind of photo switching effect that when we turn it on again, some population of them will go from the dark state back to the bright state. Another thing that is helpful here is that we are scanning very fast, but every time, for the same pixel, it will wait by 1 microsecond before we excite it again. So there is this actually a work back in 2006 by Stephen Hell's group called dark state relaxation. So when we talk about photobleaching, how is the molecule photobleached? There's many different pathways. One of the pathways is that when the molecule is in the excited state, sometimes it does not go back to the ground excited state directly. It will go into a triplet state. where it will stay for like a microsecond for a longer period of time. So what the Stephen Hell's group showed that if you wait, instead of after excited again a few nanoseconds later, if you wait until the molecule go back down from the triplet state to the ground state, then the molecule don't bleach as quickly. Because when they are in triplet state, they are very susceptible to oxidation because the oxygen is in a triplet state. So what they call dark state relaxation, we're actually increasing the pure number of photons you are getting. And that's what we're seeing here as well, that they just don't really bleach as quickly. So those are the raw unprocessed data, as you can see here. So we could image a cell like for 3 minutes, for example, one of the spontaneous activity cell. And we stopped the imaging just because we stopped. There's still photons coming out of those probes. So we were pleasantly surprised by that.
Do you think you can image the spikes of the projections from the LGN instead of just imaging the region?
Right, right, right.
The spikes.
Right. Well, if we have good enough sensor, we will. So right now, we are really limited by the brightness of the sensor. So there's lots of like exciting voltage sensor that are based on opsin. The problem with all the opsin-based sensors, they don't report voltage with 2-photon excitation. So we are limited by that. I'm just kind of keeping my ear open and try everybody's sensor at this moment. Yeah. Thank you.